First, we start with the double-stranded DNA sequence. Then, the TATA box is located upstream from the start point. To start the transcription process, transcription factor D binds to the TATA box, and then A and B also bind to the TATA binding protein of transcription factor D. Then, transcription factor F and RNA polymerase II are bound together, and they both bind to the transcription factor DAB complex. Transcription factor E and H contain helicase and protein kinase, and they bind to form the pre-initiation complex. ATP is added and then phosphorylated. At this point, RNA polymerase II begins transcription by moving along the DNA and opens up each strand so that the 5' to 3' RNA strand can be coded using the complementary base from the DNA. We are left with one strand of RNA, and this is called the mRNA, which is used to code 4 amino acids. The 5 cap is added along with the poly A tail at the 3' end. Introns, which are non-coding sections of the RNA, are removed, and we are left with exons that join together, which is called splicing, and form a functional RNA sequence. Once we have a functional mRNA sequence, the mRNA travels to outside the nucleus into the ribosome. While the mRNA is traveling outside the nucleus to prepare for translation, the amino acid and ATP bind to the active site of aminoacyl tRNA synthetase. ATP loses pyrophosphate that is hydrolyzed into 2 phosphates, leaving A and P. tRNA binds to the aminoacyl tRNA synthetase, and the active tRNA releases from the enzyme to the ribosome. The mRNA feeds through the mRNA binding site 5' to 3'. The start codon will start in the A site, where the tRNA is binding the start codon with the corresponding amino acid. The start codon and corresponding tRNA with amino acid move into the P site, where the amino acid is dropped off. At the same time, the next codon up is pairing with its corresponding tRNA and amino acid so it can be ready to move to the P site. The start codon reaches the E site, where the tRNA leaves the ribosome and back into the cytoplasm. Each codon, as you can see, will go through the same process as the start codon until it reaches the end codon. Once the stop codon reaches the A site, the release factors bind to the A site with GTP. This then triggers the release of the polypeptide from the peptidyl tRNA. The ribosomal complex comes apart and is used for the next polypeptide. Now we have a polypeptide chain that will be spliced and put together to make a protein.